So with that, I'm going to introduce you to Andre Garcia. And Andre got his PhD at the University of Georgia, working with Daniella. And also did a postdoc there as well. 2.5 years ago or so, he took a position with Angus Genetics and AGI and has been working there ever since. So with that, I'm going to turn the podium over to Andre and let's give him a welcome.
Morning, everybody. Thank you for coming to this session and joining us today for this presentation. Again, I'm filling in for Dr. Daniella Lourenco, which are big shoes to fill. She relayed all the slides and all the material prepared to me, and so I'll do my best to convey all the information for our discussion this morning. And so, as the title of the presentation goes, so we're going to talk about some new technology that are, either going in or things that are investigating now to be able to incorporate some new information in our genetic evaluations, right, to predict our EPDs. And so, to get started, right, the first part of this presentation, we're going to be an overview of what we currently do and what we've done, the advancements that we've incorporated into our genetic evaluations with genomic information, right? And so to get us started, how about we talk about how much information do we have in our current genetic evaluations, right, in our current genomic predictions. And so if you look into this slide, right, and this is an example from the genetic evaluation from the American Angus Association, we currently have about 9.6 million birth weights into our genetic evaluation, a little over 10 million weaning weight phenotypes, and a little over close to 5 million yearling weight phenotypes, right? And then we have other traits which are some of our carcass and ultrasound records, which you can see there, and some of our newer and more expensive to measure traits like feed efficiency, where we have a relatively smaller number of records, right? But nonetheless, there's a lot of data that are being included into our evaluations, but in other industries as well. A lot of information going into calculating EPDs and genetic evaluations. And so one of the things that is interesting with genetic evaluations is we're trying to predict, right, the genetic merit of the future individuals. And so prediction is typically very hard to do. However, we've seen over time, right, that if we have good enough data and a lot of data, and we use the right statistical tools and models, we can do a pretty good job. And Daniella put together this really nice example right from the poultry industry, which I think goes to show the impact that genetic evaluations have had and still have, right, on across the industries. But I think poultry is a very interesting example. So if you look into this picture, right, you can see a broiler line from the 1957, how those animals look like. And then if you look into that bird on the other side of the picture, it represents a selected line from 2010. And so you can see the contrast between those two birds. And of course, the one in the 2010 probably relates more to what we see in the grocery stores today. And so if you see the title of that slide, that says pre-genomic EPDs. And I think it goes to show that we've been doing selection for a long time, right? And that it goes to show, we had some discussion yesterday about, a business case for EPDs. Right, so this is a great example to show us in practice that genetic selection works, and when we collect good data and act on it, make selection decisions on it, we can have great levels of improvements in our population. And so, of course, nowadays we use genomic information for higher accuracy in our EPDs, and we'll talk a little bit about that as well. And then, what are the new technologies that we can still incorporate and hopefully improve our predictions even forward, so that we can keep pushing the genetic gains across our populations. And so here's kind of the mission, right? Our goal is always to keep improving. And so we had genetic evaluations based on pedigrees and phenotypes, and we incorporated genomics. Now we have pedigrees, phenotypes, genomic information. We're looking to what is the next thing that we're going to incorporate into our genetic evaluations to keep improving. I think it's a common theme at BIF, right? We know what the mission are. We're all being, asked to produce high quality protein across many different species with, animals that are more resilient, that are disease resistant, that are more efficient. And so we have to take many things into account. And so heard a lot of the discussion about environmental sustainability and all those kind of things, but also, we have to put together those production environments such that those animal production systems don't compete for water resources and for food resources that we could use for humans. And so there's a lot of pressure in the ag industry, right, for the whole food production segment. And so it's a very important mission, I would say. And of course, if we think about the context of genetics and what kind of technologies we use, genomics came into a really good timing for us to help us improve our predictions, right? And so a few areas where genomic information helps us, of course, here I have a picture of a Manhattan plot from a genome-wide association study. So it helps us identify regions of the genome that impact economically relevant traits. Another impact that I think is very beneficial and coming from a breed association perspective, right? Good identification of pedigrees, right? With progeny testing. And so our pedigrees are more accurate than ever now because of genomic testing, right? We can test those animals, verify those parentage, those pedigrees. And so, of course, here's a representation. A lot of the ways we use genomic information to our genetic evaluations to account for the realized relationship between individuals, right? We've always used the pedigree information for that, but genomics adds in another layer of information. And so why is genomics helpful, right? And hopefully most of you in this room are familiar with this idea, but this is always a nice equation to show. It's simple enough that you can explain it to many people, but I think it's interesting. So this is the equation that predicts the genetic gain, right? Sometimes it's called the breeder's equation. And so you can see many components in that equation, right? So you have the I for the selection intensity, you have the R, which corresponds to the accuracy of selection of individuals, the genetic standard deviation for the genetic variabilities we have in our population. And then finally, the L is the generation interval. And why this equation is important here? Well, when we think about using genomic information in our genetic evaluations, we're targeting two specific places on that equation to improve our rate of genetic gain. And the first one is the R for improving the accuracy of our genome or of our predictions, right? We can select animals in a more accurate way. And the other one that's very interesting is the generation interval. If you were at a presentation by Filippo Migliore yesterday, I think he showed a great example of what genomics has done to the generation interval in the dairy industry. And that is a great contributor to accelerating the rate of genetic gain in those populations. And so these are some examples of where and why adding genomic information help our predictions. So why does it work, right? So first, we're used to adding phenotypes and pedigrees into our genetic evaluations. But if you think about it, when we add genotypes, we're adding an extra source of non-redundant information into our predictions, right? So we're able to capture more information about our populations and the animals we're going to select by using the different sources of information. And so hopefully by the end of the presentation, we're going to add a few more of those technologies that could be potential sources of new information that we can consider into our genetic evaluations to further improve our prediction accuracy. And so here's another example where we're showing, we're going to show a few examples here in different species as well, but changes in the rate of genetic gain, before and after implementation of genomic selection. And this would be a graph that Kelly Ritalik, the president of AGI, has put together a while ago. And this has basically, if you look into the y-axis, we have a standardized unit of genetic gain. Right, and then the red bars here, we have data from 2001 to 2010. So basically quantifying the genetic gain in that period. And then the grayish bar there, we have from 2010 to 2021. So if you think about that period of time, you kind of quantifying, a little bit before implementation of genomic selection and after. And you can see overall this graph, those for some of those traits, right? You see for weaning weight, carcass weight, marbling rebuy area, those gray bars are taller than the red bar, which means we've made a faster improvement, right? We have more rate of genetic gain in the past few years compared to what we had before. Now, if you look at milk or the weaning weight maternal component and the fat, right, you might say, well, those bars don't look right. But it's because it's the difference where the direction of the selection goes, right? So if you think about the milk of a cow or the weaning weight maternal, sometimes the selection, there's a downward pressure in that, and the same goes for fat. So we're still making more genetic progress. It's just the difference where we're selecting animals for those particular traits. But it's an example to show, the benefits of adding genomic information into our predictions. Another example here for those of you who are familiar, PIC is a big improvement company. If you're familiar with the UGA group, they work with a lot of species, which are, it's very nice to show examples across the boards, many different traits and species as we talk about animal production. So this is a graph. You can see the rate of the genetic trend basically for an index that they have for their selection. And you can see a clear spike up on that genetic trend after the implementation of genomic selection. around 2013. Another interesting one here is one of the important traits for the pig industry, right? They want to select for having more piglets per litter, right? Increasing the number of animals. And then the consequence of that, there's a negative correlation with the birth weight of those piglets, right? There's only so much space in there. And so as you increase the number of piglets in a litter, the tendency is for the birth weight to decrease, and that leads to mortality and some other issues. And so that's been a problem for selection that we've been trying to solve, right? And if you think about this graph, looking at that bar in the middle, that says where genomic selection was implemented, you can see we've been able to break down that antagonism a little bit and make progress in the good direction for both of those traits. So could be another benefit of genomic selection, right? We make faster genetic gain, but also might help us break down some of the genetic antagonisms that we have between some of our traits. Moving on to the dairy industry, and we heard quite a bit of the dairy industry yesterday as well, some great examples, but this is a work that would have been done by Fiona Gwynna. She was a master's student of Daniella, and she looked into genetic trends, right, similar, trying to quantify the impact of genomic selection in the dairy industry. And you can see on this graph here is the genetic trend for the hosting. I think this is, fat yield. Some of the traits, and you can see around 2009, 2010, the clearly distinction between the genetic trend, how it takes off much faster rate, right? And I think we heard from Filippo a similar story in the Canadian dairy industry yesterday as well. So you can see how the trend has accelerated since we started incorporating genomic information into the evaluations. This is another graph which I think is very interesting. We talked about, increasing prediction accuracy and decreasing generation interval. We also heard about this yesterday, but here's another example where the generation interval on the sire side or on the dam side, right, decreased significantly in the dairy industry. And that happens because with genomic information, we can make better selection or selections with a higher level of accuracy on young bulls that allows us to make selection decisions early in life of those individuals. And so that's how we decrease the generation interval, and that contributes significantly to the rate of genetic gain. Still talking about the dairy industry, if you look at this slide, we have two graphs. One graph is the hosting graph we saw in the previous slide, and then another one is the Ayrshire graph. And if you look into these two breeds, right, you can see the hosting has a clearly change in the genetic trend, right? But then if you look into the Ayrshire graph, you say, well, that case is not as strong. And so I think this is also an interesting one to show, and there's two main distinctions here, right? So when the technology was adopted, right, I think Ayrshire implemented genomic evaluations a little bit later, right? I think they're running that evaluation with genomic information since 2013, but compared to hosting, which were, kind of pioneers in implementing genomic information in genetic evaluation since 2009. The other piece is how much data is in there, right? So the hosting association or the hosting evaluation at the time of this graph, right, about 5.5 million genotyped animals. I bet this number is outdated now. I bet CDCB has added a couple million genotyped animals in there already. But if you look into the Ayrshire, only 16,000 of animals genotyped. And so, of course, the benefits of the technology depend on the rate of adoption, right? So we have all these great technologies, but it's very important that we keep in mind that technology by itself does not make genetic progress. And many of you in this room are probably in attending BIF are breeders. So you are the ones who are actually realizing the genetic gain in your farms, right? And so we have to come up with the technology, make it useful, but also implement it and apply it and actually make those mating decisions so that the genetic progress can be realized in the field. This is a slide also, this is from the American Angus Association to represent the uptake of genotyping right in the industry. I think this is a very nice graph to represent how much effort our breeders have put into genotyping their animals and incorporating the technology in their operations. So as you can see, of course, in the beginning, very few animals were being genotyped. Nowadays, we have about 1.3 million genotyped animals into our weekly genetic evaluations. And so this goes to show the adoption of the technology and how they've been using this technology in their daily operations. For a perspective, right now, about 65% of the animals registered with the American Angus Association are genotyped, right? And so that's very beneficial for us in terms of making genetic progress and helping our genetic evaluation, but also is a great resource for us to do research and come up with new ideas and new technologies. And of course, I've been talking about all the benefits of genomics, right? And I know this is there's always this debate, right, in the beef industry, and now everything is about the genotype, everything's about the genotype. But I think it's always important to come back, right, that phenotyping is still very, very important, right? So the genomic information only has the benefit it has because it has the database to back it up, and we keep collecting the phenotypes to sustain that accuracy over many generations. Myself and Kelly, a while ago, we put together this kind of exercise, right, where we thought, well, how about if we, let's say in 2010, we started genotyped animals and we say, we're not going to ultrasound animals anymore. We're going to stop collecting phenotypes. We're just going to use the genotypes in the genetic evaluation. And you can see on those graphs there, we have a group of bulls, right, where we measure the accuracy of their EPDs and see how that changed. And you can see how big of that decrease is, which means if you keep genotyping the animals, but you forget about phenotyping, you stop phenotyping, the accuracy of your selection and the accuracy of your individual EPDs drop very quickly. So it's very important that we keep that in mind, that we need to keep phenotyping the animals so that we can maintain that prediction accuracy of our prediction, but also so that we can keep characterizing and measuring the progeny performance of our individuals in our populations. Daniella has this great analogy, which I like very much, and I think it's very useful for us to understand that. She puts into terms of, using the credit card and using cash, right, and having cash, right? So credit cards are very nice, are very useful, right? Sometimes they allow you to buy stuff that you don't have the money right now, but they are only very useful if you can come back and pay the bill later on, because if you don't, you're in trouble. And so with the genotypes and phenotypes is similar, right? So genomic information allows us to make selection decision on young candidates with the decent level of accuracy. But as long as we come back and we provide the phenotypes on those individuals and on their progeny down the line, that's how we keep the cycle going. I think this is a great slide to show also the benefits of using genomic information. And I was fortunate enough to do my PhD at UGA as well, where I had the opportunity to interact with many different industries, right? And so we can have a perspective across different industries. So what this graph will have is, when we look into the prediction accuracy of our models, right, we have the blue bars that you will see in the graph in a second. Those would be traditional genetic evaluations, so only using pedigrees and phenotypic information. And then on the green bars, you have a single-step GBLUP evaluation, which is a genomic evaluation, right? And so when you look here on the x-axis, you will see examples of Holstein, Angus, pigs, chickens, and some fish. And so when you look across the board, right, if you pull all the graphs for the Holstein there, you can see the increase in prediction accuracy of those two evaluations. And of course, all the bars in the green are taller than the bars in the blue, which means the genomic evaluation has higher prediction accuracy than the traditional BLUP evaluations. If you're going to look into some of our data at the American Angus Association, right, that's always true for those different traits as well. And then we'll go into the pig industry. We have traits like litter size or number of stillborns and things like that. And then you go into chickens and fish. So basically here, we don't need to talk about which specific species, which trait they're using or not. The overall big picture here is across species and across traits, the genomic selection, the genomic evaluation has a higher accuracy than the pedigree-based evaluation. And so it's a continuous case for the use of genomic information and goes to show how much more accurate our GEPDs are compared to our EPDs from just using pedigree information. Now that we've covered all this, right, and hopefully this information was just a refresher for the people in this room, right? Now we're going to talk about what are the new technologies, what is coming up next for our genetic evaluations. And so here Daniella has listed the field, right, that we have people that have been working on and that are potential good technologies that we're going to be able to incorporate or at least investigate and see how can we incorporate that information into our genetic evaluation. So you have their whole genome sequence. You have phenomics, which would be information about sensors and cameras. We heard quite a bit about this just this morning. We have gut microbiome information, blood work, which would be metabolomics or metabolites. And then you have what's called enviromics, which is kind of precision breeding, trying to characterize the environments as well. So the goal, of course, is to have either more evaluations or more accurate evaluations for things that we already have, right? And with the ultimate goal to improve farm and animal populations overall. So here we have some references of, papers that we have very smart people working on these different areas. And so we're going to talk about each of these individual areas and see where we're at and what are the perspective for the future. Daniella put this phrase here, which I think it's a very nice one, right? Whenever we have a new technology or something to generate new data, we need to make the most of it, right? And so go after it in the implementation of that technology. How can it be useful for either genetic evaluation, management decisions, and all sorts of applications? And so we need to make the best out of it. And so this is the list of the things we're going to cover here, right? So we're going to start with whole genome sequence data, then we're going to talk about phenomics, the microbiome, the metabolomics or the metabolites, and then the environmics at the end. So whole genome sequence is not necessarily brand new, right? Since we start talking about genomic selection, we've been hearing about whole genome sequence data, right? Initial studies with genomic selection implementations, right? Looking into simulated data, they had predicted we would have gains in prediction accuracy over 100% in some cases. Now, as we started collecting data, and I just showed you the slides with the gain from pedigree to genomic evaluations, seen that those gains seem to be a lot more moderate than what we would have predicted with the whole genome sequence data or with simulated data. And so the idea was, okay, maybe it's because what we have on our current chips or 50K or the 100K, we don't have enough SNPs in there that would need more coverage of the whole genome, right? So instead of using 50,000 markers, when you look into the whole genome sequence data, you would be talking about millions of markers. And so that's what we've been trying to develop in the past few years. And we had some of that discussion in one of the afternoon sessions yesterday as well with Troy Rowe. Did a great job presenting about the use of whole genome sequence data. And so as the technology gets cheaper and we're able to grasp this data, we start to see some of those results in some of those applications. I think also part of that discussion we had yesterday as well. So far, what we've seen to incorporating the whole genome sequence into our genomic predictions, we haven't been able to make very good progress or to push prediction accuracy much higher than we already have. There's a few reasons for this, and we're not going to go into details here in the presentation, but this is an example from a big data set that was work done at UGA as well. But basically, they have thousands of animals that were sequenced or imputed to sequence, right? And they tried different methods to incorporate those data sets into genomic prediction. And you can see here on the bars, it's comparing basically on the blue, just the cheap traditional predictions we have with some sort of data extracted from the sequence data. And we don't see a lot of benefit, right? Usually those gains in accuracy with adding sequence data to our genomic evaluations go from no benefit to about 5 or 6%. That's what we've seen in the literature. Now, that doesn't mean we don't need whole genome sequence data, right? I think Troy presented yesterday, there's a lot of exciting opportunity with whole genome sequence data for research and maybe developing new models for our genomic predictions as well to make better use of the data. So if you think about genomic predictions, we could still use whole genome sequence data to identify variants, and that would even help us design new SNP panels, right? But we can also use that to enhance our GWAS studies, to work into a little bit more of the genetic architecture of our traits, find new mutations, trace problematic haplotypes and things like that. And I think, of course, as we see the price of whole genome sequencing decreasing, those applications will become more useful. And so I still think there's a benefit, especially if you're looking to research, right, to keep sequencing animals. And I think there's going to be a great source of information to find new things on the genomes of the animals we work with. So the next technology here, and this is where the territory where we might have some more cutting edge things, and some of it we heard this morning as well. But when we talk about phenomics, or some of you may have heard of this as high throughput phenotyping, right? A lot of this is based on computer vision systems, but basically we're incorporating information from sensors and cameras and all kinds of gadgets that we can use, accelerometers, et cetera, to measure and record some kind of data on those individuals, right, or on individual animals. And I think the really interesting thing with this one is that we're collecting data 24/7, right? Those cameras, they don't go to sleep. We can harvest a lot of data on those individuals. And so we could try to monitor and predict things like feed intake or grazing behavior, try to monitor fertility or ester detection and things like that. We also have things that could measure, gas emissions or temperature, humidity, and all those sorts of information. And so I think there's a lot of potential benefit here. I think I heard Juan Pablo Stivel say this morning that they can collect a lot of data easily, take a lot of pictures easy, but the processing of those data, I think that's the challenging part that we're working through. But we have way smarter than me people working through this, and so hopefully soon we'll be able to incorporate some of that technology into our daily operations as well. So it's not necessarily all new information, right? We've had some of this technology for a while, but of course, computer science and engineering have allowed us to extrapolate and extend the use of those technologies. This is a paper by Ignacio Mistal quite a while ago, where he was trying to predict basically the carcass traits from pictures of cows, right? So we've probably made strides now technology-wise and computer science-wise so that we can improve on those results. So here's an example from the group of Guilherme Rosa and João Dória from the University of Wisconsin-Madison, where they're using camera images, right, try to monitor growth and development in beef cattle. So you can see here they use cameras and RFIDs antennas to kind of monitor the animals and try to predict the growth and monitor the growth of those individuals, which is very interesting. You see there, looks like some of this research is published already, so you can look at the papers. They've been able to predict animals or predict that phenotype with 0.89 to 0.96 accuracy, which is pretty good, right? Of course, as the technology matures and we're able to train those models better, there's probably room for improvement into there. So another example of what they're doing, they're using 3D kind of body images to predict the shape. And so if you think about carcass traits, right, we've heard a lot of the discussion about beef on dairy and trying to predict some of those carcass attributes. They can use those 3D images, and I'm not exactly sure how they are set up to measure that, what the technology is, but it's quite interesting, right? And if you can use that information to predict those phenotypes, right, it could aid into our genetic evaluations, or it could aid information as in the form of new phenotypes to help us predict for better carcass outcomes. So is using phenomics a reality right now, right? So as we know, a lot of those phenomic applications, again, I'll refer to the presentations we heard this morning, right? They rely on machine learning algorithms, so artificial intelligence. And so basically those algorithms, you feed all the data through those algorithms, train the machines basically, and then they can come up with a prediction at the other end. Of course, the limitation here would be, labeling that data and making sure we're getting accurate prediction out of those data sets, right? It's very important and it's a big challenge. Of course, it requires a lot of infrastructure, right? And I can hear kind of coming from a breed association, I think we need to think about the producers, right? All those cameras, and also if they're data hungry, we need network and we need data connectivity, data storage, right? To be able to implement those things at a farm level, being able to communicate all that information. It's also, it's expensive to teach the machine, right? Those machine learning algorithms, they are data hungry, they are computer power hungry, right? It takes a lot of effort to do that. But I think it's, as the technology matures and we have more of those data sets built up, it will be able to improve more and more. And of course, computing limitations, right? We always have either improvements in the statistics and the algorithms behind it so that it can optimize the computations, or we have, sheer hardware improvements that will enable us to implement these technologies in the future. So as far as where we are, right, so we're collecting a lot of this data. There's quite a lot of research being done on this area and trying to collect data, train those models and see what kind of predictions we can get out of those. Some of the implications, right, we might get new trait definitions over time, or we might get new traits that we're going to predict over time that using phenotypes coming out of those systems. The next technology here that we're going to talk about is microbiome information. There's a lot of research being done here. This is an example from Jefferson Lourenco. He's also a professor at UGA now. So his group, they're going out and collecting microbiome samples from individuals, right, and trying to make use of this information. There's a very short clip, we'll see if it plays here, of their team trying to get a sample of the microbiome. So basically, it's a very short clip, but basically what they're trying is collect a fluid, ruminal sample of those individuals. once you do that, you can take it to the lab and then you do a DNA analysis basically. So that allows you to characterize what kind of microbes you have in that ruminal fluid, and not only what kind, but what is the balance look like? What kinds of microbial kind of contributions they have into that rumen environment. So they are looking into this information, right? And it has some results here that are unpublished. I'm not going to go over those, but basically they're trying to get this started, right? Collecting some samples. Basically, the idea is, can we associate the microbiome composition to some of the traits that we know, right? And you might think of, well, maybe we can tackle feed efficiency, or we can tackle methane emissions. And so those are the ideas that they're trying to investigate now. In fact, we're collaborating on a project where they're going out and sampling some of the herds, right? They have about 1,500 animals sampled for those microbiome from rumen fluid. And so they're currently now in the process of sequencing those samples. And so later on, we're going to try to do associations with some of the traits that we already have reported and see what we can predict out of those. So is it a reality in where we are now, right? Again, collecting a lot of this data and starting designing those experiments so we can take a look and investigate how do we use this information? Is it useful for genetic evaluations? Is it going to be some other applications that we're going to be able to use? I think a very relevant point, especially for this audience, would be, okay, how much does it cost? Can I do it easy enough, right? That's the good thing with genomics. You can, snip a hair sample, a blood sample, or an ear notch, and you're done. You have a DNA sample. Some of those others are a little bit more complicated. So for these ones, they were running about $40 a sample, and then you have to have some people trained to be able to go and collect those samples, right? So some of those are more complicated than others. Again, some of the implications, new traits potentially for genetic evaluations or association with some of the traits that we already have. Some of those implications will probably need more complex models, right, to model our genetic evaluations, some different statistical models, and that has some implications from a practical standpoint as well. So the next set that we have is some metabolites, right? So that metabolite profile, and we have here there's all sorts of intermediate omics data that we're not going to go into detail. I don't think I even could go into detail if I wanted to, but so we're going to keep it to the overall picture here. But basically, you can take a sample of the animal, right, and you can measure several different metabolites on those samples. And so we're also trying to investigate what can we do with those samples, right? So it's relatively cheap compared to some of the other technologies that basically we're trying to investigate how can we use that information for our genetic evaluations. That an index of a combination of those metabolites that will help us explain a phenotype. Is it a correlated trait that we're going to add into the genetic evaluation? Is it some sort of similarity matrix or relationship matrix across individuals that we're going to come up with? Here to cover an example of a project that they're working on with PIC as well. Again, basically they're looking into how do we model those kinds of data sets. So coming up with statistical models to incorporate metabolites, functional annotation, and sequence data into predictions as well. So again, that same slide, is it a reality where we are? Again, collecting a lot of data, trying to come up with those design experiments so that we can harvest some of that data, collect that data, and try to make sense of it, model it in a statistical way that can be useful, right? This could be, again, new traits in the genetic evaluation, or it could be an aid to the genetic evaluations we currently have. Again, probably would involve a lot of computations and more complex modeling of some of those data sets, right, to be able to extract useful information. And the last one that we're going to cover here is enviromics, right, or the precision breeding type of data. As we know, of course, for beef production, if you take, Canada or US or Australia or Brazil, big countries, big production systems, but they have different environments within country, right? And so basically, how can we make sure we model that correctly and be able to match animals to their environment, right? And so the group from Wisconsin, led by Guilherme Rosa, have some, I think some study here done by them, trying to work really hard to capture that information and try to help us model G/E. And of course, you have headlines like this pop up and then everybody's concerned. And I think, we need to make sure we work the best that we can to use the data that we have to try to model and explain and match those individuals to that environment, right? So we have our resources in our population. How can we make sure that they are allocated to the best that they can be in different environments that they have to perform? And so this will potentially help us to model G by E, right, which is a challenge to do in the beef industry. And so this type of data, right, the enviromics could help us do that. And so right now we're working on a project, and you can see it, all the pictures there, a bunch of very smart people are working on this. So try to use the data that we currently have in our genetic evaluations with data from other sources, right, from sensors, from climate stations and things like that, to be able to predict some of that information. So again, this is more of a reality in the plant breeding world. In animal breeding, not so much, but I think we're at a stage where we can collect some of that data. There are some applications for the THI index, temperature and humidity index, being used in dairy evaluations or some pig evaluations, but it's not really a reality across all the species. And so I think there's a lot of opportunities collecting a lot of that data and make sure how we model that data and how can it be useful for genetic evaluations. Heading towards our final remarks here. We covered in the first half of the presentation how we've been using pedigrees, phenotypes, and genotypes. I think that's well-established technology, right, for genetic evaluations. And then we have these new technologies, new sources of information, and I think this is where the research is at now, and kind of trying to come up with how can we best use those new technologies, those new sources of information to help us in our mission of improving genetic prediction and improve animal production. So some of the final remarks, right? The goal still remains the same, right? We want to help breeders to make more accurate selection decisions, right? And we're going to use all the technologies at our disposal, right? Make sure that they are easy to implement. They are cheap enough that breeders can access and use in their daily operations, right? But the goal is to improve the accuracy of selection decisions. There are some implications with the new sources of data that we've talked about, right? So more data, and some of those are more complex data, means there are more computational challenges. That was very interesting about this sentence that Daniella put there, right? In the beef industry, weekly genetic evaluations are a common currency now. And then as you talk about those very complex data, very data-intensive applications, can we crunch those on a weekly basis in the future, right? I think that's a very valid question. And then for the breeders, right, I think this is very important. You know, can we get this technology cheap and applicable enough that the breeders can use in their daily operations? And so hopefully some of those will mature and become a reality in the future so that we can use those information to increase genetic gain. And so with that, I'm happy that you guys stuck around for the presentation. Again, those are very big shoes to fill. I hope I conveyed all the information Daniella wanted to, but appreciate you guys, and I'll be happy to take any questions. Thank you.